recent news, we have been observed that Gujarat has been the first state to launch the tribal genome sequencing project. Hello everyone, this is Pratyusha. Welcome to Laexcellence. In today's class, we're going to discuss about what is the genome, what is the genome process that is being present. Then we'll also discuss the Genome India project that has been there, and also we'll discuss what is this Gujarat's tribal genome sequencing project that is being present. First, what is the genome that is being present? So when we see the genome is like a blueprint, or it's an instrument for the manual of the life. Now every organism has a genome which is having genes containing different sequence of the base pairs that have been there. We all know we have 4 different base pairs for the DNA, that is A, T, C, G that is being present. Now this, in human genome, there are around 3.2 billion DNA base pairs are being present. It indicates that the large amount of genes are being expressed in this genome. Every single activity of the human being is being coming from the genes that are being present in the genome. Now when we come towards the genome sequencing that's been there. Now this is an emerging technology that's been present where the whole gene is being taken, an organism is being taken. From this organism, we are removing the genome. From this genome, we have been taking the DNA. As I told, the DNA of the human is around 3.2 billion base pairs have been there. Now when we see the process of the sequencing of these base pairs that have been there, first the organism is being selected. From this organism, the genome is being removed, or the DNA is being separated. Now this DNA is being cut into smaller fragments because the length is too big. Now this fragment is being cut into small particles with the help of molecular scissors or sonication that is being present. Now with these fragments are being taken, and we will be creating an PCR, that is polymerase chain reactions. With this, we will be duplicating the DNA fragments that have been there. Now, all these fragments will be kept in the DNA library that is being present, that is the DNA read that has been there. Now, all these little bit fragments that have been there, which have been read and which have been sequenced, will be combined together, and a long sequence is being made. Now, this is what we have, the sequencing of the DNA that has been there, or else it is also called genome sequence technology that has been present. Now, in India, we have already successfully completed the Indian Genome project that has been present. Now, it has been launched in 2020 by the Department of Biotechnology under the Ministry of Science and Technology. Its main aim is to map the genetic diversity of the Indian population, and it is to create a repository of the data that is being present. Now, this Genome India project was being led by Center for Brain Research, IISc Bangalore that has been there. Now here we have taken around 10,000 of Indian genome has been collected from the individuals across the population, across the races that have been there, across the different religions that have been present. And we have created a data set of 8 petabytes that have been there, and this data has been actually stored in the Indian Biological Data Center, Faridabad, that has been present. Now when we come towards the Gujarat's tribal genome project that has been present. Now Gujarat has been the first state to establish a tribal genome project that is being present, where it is being led by the Gujarat Biotechnology Research Center. Here we have sequenced the 2,000 individuals from 20 plus tribal groups across 17 districts that have been there. Here the participation or the selection of the individuals plus be more than 18 years, and here we are taking 2 things. One, we are seeing for gender parity, and the other one we are going with the gender trio sampling. Gender parity is that we are across the 20 plus tribal groups that have been there, the male, the female that have been present. We are trying to gather the DNA samples from them. And here we have gender trio sampling, is where we have been to sequencing the mother, the father and the child. Now this is where we want to see where the genetic disorders are being hereditary from the parental to the child that's been present. Now what this genome project, or why or what type of benefits are being present with this genome project is that, one, it will help us to understand the varied genetic diversity that is being present in the indigenous tribal population that's been there. Now one thing is that in our tribal population, they follow the endogamy that's been there. This is where they marry the people of the same community that's been present. It means that the gene that is being present is being present in this one. There is no marriage outside has been seen in these tribal groups. Second, it can increase the disease prevention. Now when we know this particular sequence of this gene is being causing the genetic disorders, for example sickle cell anemia or thalassemia that's been present, we can easily alter the genes and go for genetic engineering techniques. Next, we have new insights to human evolution. Now we can link why these are different tribal groups that have been there, how they have been evolved, what is the ancestral things that have been there, what are the genetical disorders that have been present in this tribal group and all. Then we have unique identification. Then we have identification of unique variations that have been there. Now we can divide or we can differentiate different tribal groups that have been there, different uniqueness that are being present. Now through this thing, we can understand different genetic or through this project, we can understand different genetical disorders that have been present in the tribal groups. Thank you.